various areas, I strongly encourage you to chime in, and I think this can be a fairly lively discussion. So part of the reason that I wanted to talk about this is that I think an understanding of how exactly these data are generated can be very helpful in terms of understanding different error modes, in terms of understanding what can go wrong, and what our data actually mean. And so this is a fairly high level, fairly cartoony version of how our data are generated. And just a quick bit of definition. We had our central dogma in the first lecture on the first day. And we talked about this DNA, RNA to protein sort of cascade. And the omics technologies, generally genomics is the study of DNA on a large scale. Transcriptomics is the RNA piece, and proteomics is protein. So sort of combining across these technologies. And then epigenomics would sit here, where we're talking about methylation and regulation of transcription. And so when people talk about new omics technologies, this is generally what they're referring to. So we can measure variation in DNA. You guys all have access to a very fancy data set that has genotype information. And so this is just a little bit of background on the history of how this all came to be. So the original Human Genome Project, I imagine many of us were quite young when this was completed. The original Human Genome Project was completed in 2003, so that's still in many of our lifetimes. They sequenced one haploid human genome, so they only sequenced half of a person. They didn't sequence a whole person. Lots of people have been asking me who this person was. They put an ad, my understanding is they put an ad in the paper in Buffalo, New York, and said who wants to volunteer their genome for science, and they had a handful, like less than 20 respondents, and this one particular sample had very high quality DNA. And so that's our random citizen from Buffalo, New York. This project took 13 years and it cost $2.7 billion. And believe it or not, this project was under budget. So somebody budgeted almost $3 billion to sequence half of a person, and this is not that long ago. So the idea with sequencing is that we're going to exploit this idea of the complementary base pairing. If you have a single strand and you have an A base here, we know that if we just put a bunch of individual free-floating bases, that the T is going to bind here. And so most sequencing technologies really leverage just this complementary binding piece, and that's really the key to understanding, I think, all the technologies I'm going to talk about. So the first automated procedure to do sequencing, and this was a while ago, this is Sanger sequencing. I should mention Sanger sequencing is still considered the gold standard for sequencing. It's incredibly accurate, but it's also very expensive. And so the idea is that you have some strand that you want to sequence. You have some primer that's going to bind to that strand. And then we munch it up into a whole bunch of different lengths. Shine a laser on it. And we get binding of these different bases to the one on the end. And we label each base with a different color, a fluorescent molecule. We shine a laser on it, and there's a detector here, and it reads out what color fluorescence did we see at each base. So we digest these up. This very short segment of DNA, we digest these up, put them in order of size, and so hopefully we've got all the bases on the end. This takes quite a bit of DNA to actually get this to work. And the optical readout on the other side is these peaks of a color. This is what fluorescence actually looks like. This is a zoom in on what's actually happening here. So we have our strand, we have the primer that binds down here. We have a DNA polymerase and set that loose this way, and we look at which color gets added. So if you look at Sanger sequencing output, We don't need that. Go away. So another way to think about this is that, like, as we go through, as we chunk through, we get a little peak for each base, and they're in different colors. A nice output for this would look like this, where the peaks are really evenly spaced. The colors are very clear. I should mention some of these slides I've borrowed from Elizabeth Atkinson, who made these slides for teaching. Courtesy of Alicia, actually, is how I found out about them for the Ginger Project, which is a collaboration with Broad and some researchers in different countries in Africa. And so you would see these nice little peaks. You would be able to call your bases clearly. Sometimes the output looks like this, where the peaks are not clear. There's obviously some mix of the 4 bases, but it's very difficult to tell them apart. And as this technology developed, something that looked like this would have to be rerun, which gives you an idea of where the $2.7 billion went. Yeah.
So what was causing the problems here?
I don't think it's always super well known. I know with these kinds of technologies, the optics became issues very quickly, and the rate at which the optics could progress and keep track of the fluorescence was a thing. There were lens issues. There were lots of things, but I was not around for these days, and I am not sure.
For those days, something can like go wrong, or like your salt concentrations are off, or the amount of enzyme that you add to the tube is off, and so it's just like sometimes it's just like things that you can't anticipate that are like small biochemistry related issues.
Mm-hmm. So, for the original Sanger sequencing project, the fragments were pretty small. They were in the order of 100 bases. Sometimes there were a couple hundred, but we weren't getting fragments over 1,000 bases. And all the sequences collected, because there wasn't a reference human genome, had to assemble them. And so you could imagine if these were your sequences and you had to do this all by overlap, that it could take quite a long time and that it would be quite a large computational problem to assemble all of this. So that was Sanger sequencing. Sanger sequencing is the older technology. As I mentioned, it's still sort of the gold standard in terms of quality of sequencing. However, it is extremely expensive, and with this technology, you can sort of see why. But fast forward a little bit to SNP arrays, and I think this is probably where we are most interested, is we talked right before the break about how SNP arrays have biallelic SNPs. We really do rely on the fact that they're biallelic in this technology, and that this technology has been getting cheaper and cheaper and easier to do on more SNPs. And so the way this works is that you have 2 strands, and they have some number of SNPs on them. You have to amplify the genome, because this is usually not enough. This is another fluorescence technology. So this is the amount of DNA that you would actually get in a sample. Even a blood sample would not be enough. You have to bust up the DNA, and so it's in small pieces. And this, I think, is actually worth drawing on the board. And I should mention that the different technologies have different details in this. But this is sort of the upshot, that you have something like a microchip, something stable, and you have attached to it, you have a piece of single-stranded DNA with a very specific sequence. This is probably not a probe for an actual gene, but you have a very specific sequence. Right now, modern chips usually have about 50 bases, and it's sticking up off the microchip. So when you wash your DNA over it, some of these procedures involve heating up the DNA so it denatures a little bit. You end up getting your single strand, and usually the SNP is here, the SNP that we're interested in. You get your strand binding specifically here. So the idea is that this is your sample, this is the probe, and this is your sample. And the idea is that this matching of sequence context is what gives you the specific binding here, and not just any old DNA. So then we put some of the procedures will trim this so that just the SNP hangs off. Other ones go about this in other ways. But the upshot is then we wash over this some fluorescent probes. So you would have, these are not the actual colors, but the idea being it's usually red and green for a specific SNP, and you might, and some of these they'll have a different address for different ones, and some of them they don't. But anyway, so if your sequence had a C here, the G probe would bind here, and it would fluoresce green, and so that's how we would know that you had a C here. Yeah?
So this G probe is another additional, just one base probe that's different from the one that's already on the
Yeah. So the G is one base by itself. The probe on the chip is a sequence of some number. I think some of the original chips had shorter probes. There were maybe 25, 26, and most of the modern chips have a probe that's about 50 bases long. And so there were sort of two different ways. There were the red-green chips, or the early AFI ones, where they would put one color on one allele and one color on the other. So you would get a picture that looked weirdly holographic, where some of the dots would be more green, some of them would be more red, and some of them would be yellow. And so the more red dots would be homozygous of one allele, the more green dots would be homozygous of another allele, and then the yellow would be the heterozygotes. I think most modern chips are one color, and they just have locations for, say, this is the C allele, and then they would have another probe here and say, this is the G allele. Yeah?
So if this is the technology, it means, I guess, that you have to pick the SNPs that you're going to measure in such a way that you have different probes that lead up to them. So that's going to be a constraint on which SNPs you can measure.
Yes, absolutely. So you could imagine something where you have a sequence and your SNP is here and you have some kind of indel here. It would really mess you up because now you're not going to get specific binding here. So yeah, probe selection and the probes being functional is really, really important. And again, that is not something I work on, but I know a lot of people do. So this is another kind of a sideways version of this. This is the sort of older AFI technology where you have these are your SNPs, and you have your DNA sort of annealing to these probes that are sticking out. And then depending on the sequence of that DNA, different fluorescently labeled probes will bind. So on SNP arrays, this strategy gives you a fluorescence measure for allele A, for allele B, and from that we have to come up with a genotype call. And so these sorts of plots are really common in the genotype calling area. You would get on the x-axis, this is the allele A intensity, and on the y-axis, the allele B intensity. The colors here have been assigned afterwards, so these would just be points on a graph. Yeah?
Are the SNP arrays effective for use of different ancestries? Does it work for everybody? Or?
The calling or the probe design? Yeah, I think that's a pretty valid concern, and I don't actually know of examples of that. Alicia might. Do you know if probe design and probe specificity varies by ancestry, and if our Eurocentric bias has an effect on some of our probes?
The probes that you design are always designed in the same way, but which SNPs go onto the arrays vary, and those are pretty Eurocentric and biased, which is a big problem when you're trying to query variation from, for example, African populations, which have a whole lot more genetic diversity. And so when you look at what the allele frequencies are in those different populations, you can have these really big skews towards low ends of the allele frequency spectrum in different populations.
But could you have a problem where they're like the probes are designed based on like haplotypes that exist in like European populations, right? But let's say that
in Africa that haplotype just doesn't exist, right? You couldn't measure the SNP there because
Yeah,
the probe is
because there could be like a common African SNP here.
Helps like bring back to earlier in Andrew's presentation, like what the reference genome is. So the reference genome is a pool of a few individuals, but one individual dominated, and that individual had African ancestry. And so our human reference genome that we compare everything to is an African American individual. And so if the probe is mismatched enough from that reference genome, then it won't bind. But you have to have usually multiple variants that are different in that specific probe, which is only about 20 base pairs long. And usually that's not the case, but if you have indels or some other complex structure in that region, then it won't necessarily anneal, and then that can cause some problems.
so like little differences are okay.
Those are tolerated, yeah.
Yeah. But something big, like a deletion, would totally mess you up. I don't know that that's common, but in any case. So from this, we have to call genotypes. And so the idea being that you want this one cluster to be almost no B allele intensity and all A allele intensity, something in the middle for the heterozygotes and something the other way around for the homozygotes of the B allele. And so this is sort of how genotype calling is done, identifying these clusters and drawing nice lines in here is its own area of research, and lots of folks have worked on this problem. You might imagine that some of these near the border, you'd be slightly more skeptical of the genotype call coming from those than the ones falling right on the line. Sometimes you get something that looks like this, where every individual in your sample appears to be a homozygote. And it is very possible that every individual in your sample is a homozygote. However, this could also mean that this probe just bound everything. Or, if the scale of the y-axis changed, it could mean that your probe bound nothing, or bound some things non-specifically. And so there's not a great way to tell if this is a truly non-variable SNP, or if this is just a funky probe. And so this is part of the reason we throw these out in QC, is that sometimes the probes aren't perfect. Sometimes, and this is going back to our structural variant conversation, you might see some extra clusters in your cluster plot. And the ones that look like this are sometimes errors or batch effects, and sometimes this could be a CNV. If you see this very nice sort of radial pattern, you have 4 B alleles here, you have 1 A allele here, 2 A's, 3 A's, and 4 A's. You would see this ratio in the fluorescence. Yeah.
What I thought that what's getting attached to the probe at the end is just a single nucleotide molecule.
It is.
So how would you get the...
So we're doing this on many, many probes, identical probes. And so at the end, we look at this through a lens, and we're measuring the level of fluorescence off of this spot versus the level of fluorescence off of this spot. And so these are both continuous measures. Unlike Sanger sequencing, we're not doing it one at a time on one molecule. We're doing this in a very multiplexed way. And so the idea being that we have lots and lots of these little fluorescent Gs being picked up by this lens, and lots of these little fluorescent alternative alleles. And so that's why this plot is continuous and not discrete, because we're talking about comparing 2 levels of intensity from a fluorescence experiment, and not a yes-no sort of a call.
But so if you did the trick where you cut off the DNA and so that there's only one nucleotide above the top of the probe, then wouldn't you always be getting the first repeat of the copy number variant? Does that make sense?
Yeah, I guess you would. Unless the SNP is inside of a copy number. So suppose you have the same sequence and it repeats here, and our probe is this sequence. There'd be no reason that you couldn't bind here or here. So that would be assuming that the CNV happened like right at the SNP, which is probably pretty rare, considering that these guys are pretty big. And so this is an active area of methods development. Being able to actually identify cluster plots of this type, it's a pretty hard problem, and one of our coworkers at Broad has been working on this on and off for the last couple of years. So maybe someday we can also call CNVs from a GWAS chip. And so, as you can see, there's some molecular biology that goes on, and then there's some arbitrary calling that goes on in order to get us to a place where we have a matrix, where we have samples and covariates and genotypes. And so this can contain errors, and they can come from a lot of different places in those processes. And so that's why our QC processes are what they are. Yeah.
In the previous graph?
Previous graph. Yeah.
So, if I understand correctly, you run this multiple, same probe for multiple materials from a single person, and you get an intensity for a specific person.
Okay, that's a really good point in understanding these plots, is all of the samples are on this plot. So every point is a person. And so if we have a certain ratio of A allele intensity to B allele intensity in one person, that's not necessarily informative. But if we put them all together, or we put them together with some control data that we know what they're supposed to look like, usually provided by the manufacturer, you would end up with these clusters of points. And so it's this pattern that we're looking for. So this is all the people in the sample.
And each point comes from multiple strands like that.
Yeah. And so, on the location on the chip, there are several probes, identical probes, all in the same address on the chip. And it's the chip manufacturer's job to make sure that they know which probe goes to which genomic address.
The information about uncertainty, is that thrown away? Or is it something that is conserved and used in research?
It is conserved. The genotype chips will give you a quality score.
Per location or in general?
Yeah, it's per SNP. So it'll be per SNP, usually in the data set. So it'll be per SNP for the whole lot of them. And is that right? It's per SNP per sample.
It should be, right?
Yeah.
I'm trying to think of like the standard data sets that you would interact with. Like, if you're downloading the genetic data from AdHealth, it's not clear that the baseline data they said you would give you
The base call quality.
Yeah, I'm not
Because
sure.
we usually filter that.
ISU might know. Do you know, ISU, if the genetic, like when we receive genetic data from HRS or AdHealth or something like that, does it have a measure of like how confident they are in the genotype call, or do they do the quality control first before they give it to us?
It depends. So sometimes they do some quality control and then give it to us. Sometimes they don't, but they don't give us the measures of how much you trust in the genotyping of a certain SNP. But you can filter for call rate, I guess, because if the call rate is really low, it means that we went wrong in the genotyping of even of the ones that are not missing.
So you might know for the whole, the whole SNP, if generally there's probably, but you wouldn't know for the individual and the SNP, like where they landed in this plot necessarily.
You wouldn't know if you were this person here or if you were in the middle.
Sometimes they maybe provide it to you to like ask what these data sets are like.
I don't
They get to
think
be
so.
large.
No, I've
No,
never
they wouldn't
seen.
even be provided even if you asked.
I've gotten them when I've asked for the GTEx data. They have expression array stuff, and they did give me the raw ones.
You wouldn't get it at baseline.
But the second question is, would that be helpful for GWAS or this type of analysis to have that information?
I mean, if they haven't, so we're assuming that they have done some QC on the data and you have those data, so after that point you don't need it actually. But if they haven't done anything, if they just give you the raw data without any quality control, you don't want to include all of those SNPs in your GWAS or whatever analysis.
And I think this call rate point is really, really important. That even the manufacturers have some standard of what they will call a genotype and what they will say, it's too close to the line, we just don't know. And so if they're doing that a lot, that tells you something about how goofy this this cluster plot looks.
Yes,
Here.
that was my question. What exactly is call rate? And is it the number of SNPs that we're able to call with certain
Yeah. So the call rate is the number of samples, and you can correct me if I goof this up. It's the number of samples in your whole study that they were able to correctly, well, or they think correctly, so confidently, not correctly, but confidently assign a genotype.
So things near the boundary, like, you know, they might just tell you we don't know. And so if that's too high for a particular SNP, then you might think that there was maybe a problem with the genotype.
Yeah, and if
The ratio of individuals for whom the SNP is not missing, the genotype is not missing.
Yeah. So you could imagine a case in which this cluster plot isn't a cluster plot at all, it's just kind of a big fan. You might get a high rate of missingness because the algorithm, for whatever reason, drew some arbitrary lines, but so many of the points are too near a line. Would be sort of the way I would think about call rate. Okay. So I know because GWAS technology is the thing that you guys primarily work on, I was going to introduce next generation sequencing and some further down the line technologies, but is there anything else we want to talk about with SNP arrays? Because I know this is the data set that you guys actually have. Resounding silence. Okay, cool. Still got about half an hour. So we will talk about next generation sequencing, and this is really should be called present generation sequencing since this has been online for a while. But I think there's a difference in the language here that we should maybe be aware of, that sequencing really does talk about sequencing every single base, and genotyping is what we do when we get a SNP array, and that's some very carefully selected, curated bases in which human variation is common. And so a lot of the missing heritability question historically was addressed by, well, we haven't sequenced. We don't have every single base. And a lot of people said, well, if we could just sequence everyone, then we would answer this question. So far we have not, but that was the thinking. And so next generation sequencing, also called second generation sequencing, which I think is why it was next. The whole genome can be sequenced in a single day and for a few $1,000s. So this is much, much cheaper than Sanger. It's still much more expensive than a GWAS.
Would Sanger still be, if we wanted to sequence the whole genome, be the $3 billion from before?
No, I think it's cheaper now. I didn't look up a figure, but I'm sure we could find one. My understanding is that Sanger sequencing is still used in labs to validate targets if you want a really, really accurate sequence of something that you're really interested in, but it's usually used in a targeted way and not so much a let's do the whole genome using Sanger sequencing sort of way. So this is a high throughput, massively parallel technology, and it takes advantage of alignment. That means that now that we have a complete human genome, we can use it. And I have a slide talking about that. So this is a graph of the cost and how the cost has declined over time. So the blue line is the cost per genome, and then the red line is the cost per megabase. So if you want to do just a very targeted bit of sequencing, this is getting cheaper a bit faster. And so you can see this curve drops sharply right around 2008. And this is as this technology was being developed. Cool. So this is a little bit of a cartoon of how this technology works. This is the family of technologies that I work on, so I'm kind of excited about it. So the idea is that you have some DNA that you've isolated from your biological sample. You clone the fragments, and there are a variety of chemical ways to do that. And you sequence each of these fragments one at a time, and then you use the reference genome to put them back together. So for next sequencing, this is a zoom in on how we actually do the sequencing. So this genomic DNA, we chopped it up, we added linkers, and what happens, this is called a flow cell, and it has lanes, and what ends up happening is that your linkers, one on each side, bind to the two sides of your little bit of DNA. This also works for RNA, I should mention that. And if you bind on the blue side, The red side is very near, and so you get these little rainbows. So then the sequencing occurs from both ends. So we're sequencing the blue side and the red side, and we sequence along our little rainbow until the polymerase poops out. So we have this. This is our DNA. And these fragments can be variable in length, but they have to be in a certain size range so that the sort of mechanics of this all works out. And these guys are sequenced this way and this way. And this leads to a paired end read. So you end up having a blue address and a red address, as it's shown in this picture. And you have a bit of sequence from one side, and you have a bit of sequence from the other side.
So you don't know what's in the middle?
Well, you learn what's in the middle by alignment. Patrick asked, do you know what's in the middle? And the answer is, do you sequence what's in the middle? No, you don't. And this is the problem that long-read sequencing tries to solve, is that it tries to keep going and keep going until they cross. But at least the last I checked, the rate-limiting step on that was the polymerases were just crapping out before we could get all the way there. Yeah. So you end up with a paired end read, and this is a little bit of a strange data format, right? You know that this sequence biochemically has to have some range, some distribution of lengths, and that there's a sequence on either side. This is what they actually look like under very, very high resolution. As you have one end and you have the other end, you sort of meet in the, or you aim to meet in the middle. You never quite get there. So alignment is the fragments are aligned to the reference genome. So I talked before about how the original human genome needed to be assembled by similar sequences. These ones get aligned. And the way I think about this is, if you're doing a jigsaw puzzle, you could do the jigsaw puzzle with no information about what the picture is supposed to look like at the end, or you could just look at the box. And so assembly is what I think of as doing it blind. You don't know what the reference sequence is. Whereas alignment, you know what the picture is that you're trying to make. You know the overall structure, and you're just trying to match the pieces to the original picture.
So tell me if I'm misunderstanding. It sounds the way you're describing it, that essentially we're imputing the stuff that's in the middle of the what did you call end reads, or what
The paired end read?
The paired end read, yeah. Because we don't know what's in the middle, and we're just guessing based on the reference genome.
So yeah, we are just guessing based on the reference genome in order to do alignment, but we don't use the information in the middle when we do variant calling.
Oh, okay.
So I think I have a picture of, yeah, I do. Okay, so this is a real example of what it would look like if we had our reference genome up at the top, and then we had all of our reads, and whether they're flat or arrowed on the end. It tells you which pair in the read they are. And you can see that they overlap quite a bit. And so when we do our variant calling from this sort of thing, we're only interested in the ones that we've actually sequenced. So the bit in the middle is used in the alignment step, but it's not used for the variant calling. Yeah?
Why would Sanger sequencing be the gold standard? I mean, this sounds very good, like better, potentially better, because you are using this information from the reference genome that helps you get accuracy, and you don't have to make probabilistic guesses about what matches up with what.
So I think there are two answers. The first answer is that just in terms of the sequencing part of this, the Sanger technology is just more accurate than what we do here. And so there are single base errors that happen in this technology all the time that are just less common in Sanger. So the people will quote high error rates in next-gen sequencing and then long-read sequencing as well. I'll talk about how the error rate is higher, and that's specifically what they mean is the base calling error rate is higher in this technology. And then the second thing is that if you're interested in a region such as MHC or something that has a lot of chromosomal rearrangements. So something that the reference may not be that great of a reference. And the MHC is a hit for schizophrenia, so we can talk more about that if we want to. But the idea, yeah, we might as well. So the MHC region is a region of chromosome 6 that is involved in immunity. So if this is chromosome 6, kind of right in the middle, and it's a fairly long swath of the genome that contains several genes. And this region is highly mutable. So when we talk about a reference sequence, I think there are 12 MHC reference sequences now on UCSC. So it's a highly variable region. The idea is that diversity in this region is highly selected for. Immune diversity is valuable. The idea that this is the bit of the genome that encodes for things that make antibodies and that sort of thing. So the idea is that this is largely the same across people, but the MHC is really, really different across people. And I use this as an example because I had a project in graduate school that asked me to align and then de novo assemble the MHC, and it was too hard of a problem for the technology at the time. So there are regions of the genomes where you might want to know the exact structure, and you might want to know exactly what's going on in terms of the order of things. Like genes can change order in the MHC in different haplotypes, which is unheard of in the rest of the genome. So you might have MHCA or HLA and HLA-B might swap locations. So that region, if you wanted to figure that out, you might want to do something more long read E than alignment. So it's the two bits of answer to that. Yeah.
So where is the reference genome from?
The reference genome. Okay, so the reference genome gets added to all the time, but the original reference genome was this haploid genome from this random person in Buffalo, New York. And as Alicia just mentioned, the only things we know about him is that he responded to an ad in the paper in Buffalo, New York, and that he appears to be imputed to be of African American descent. And that's really all we know. That's all I know. I'm sure someone knows his actual identity, but that is not public information. So it's some guy.
But would it matter if then the race and ethnicity of
It might matter a little bit, and so as more people got sequenced, the human genome, the reference genome gets updated. So when you talk about the build of the reference genome, that's what we're talking about. We're talking about the region. And in that build is included different haplotypes on the MHC. They actually show up as their own chromosome because they're so different if you look at the annotations. And information about which loci are SNPs, where known repeats are, where known CNVs are, all gets annotated into the reference genome. And so the reference genome grows as we get more information.
Yeah, and there was like a very long intercontinental effort to basically adjust for those variations, and still there is this debate now that even the adjusted human genome is still skewed towards certain populations but not others. And with long reads, there are like very recent papers in Nature Genetics about this skewness, and they're using long reads to hopefully adjust it further.
Yeah. So the reference genome is not as much of a reference as the name implies. It is still a moving target. I think we're on build 38 now, is the current. But so if you're using a piece of technology and asking you what the build of the genome is, they're talking about that's important for getting the address, the specific address of your SNP or the specific address of your gene to match up with the current reference. The sequence is not changing, but the way we annotate it is. Okay. So coverage and depth is another important concept in this technology. So the number of independent sequencing reads that cover a particular nucleotide. You can imagine that if we took our whole genome and we busted it up into little fragments of sequence and then did this to them, the number of times we did this is a little bit it's like the number of random draws from the genome in terms of what location we got. And so coverage is measured in, on average, how many times did you sequence a specific base? And that does refer to the ends and not the middle. And so project's coverage ends up to be around 30x is considered pretty good. Low passaging of a whole genome would be something around 5. That would be considered very low coverage. And you can get higher coverage if you want to be more confident about the calls that you're making. So higher coverage does mean better data, but it also means a lot more money. And so then you have to decide how you're going to spend your money. Are you going to spend it on higher quality genotyping, or are you going to spend it on genotyping more people? Yeah.
Might be worth saying something about especially low coverage sequencing, like 0.3 sequencing, because some of the data sets in the social sciences, like the child development supplement of the PSID, has gone with especially low coverage sequencing.
Yeah.
Rather than genotyping SNP arrays.
Yeah, and I don't know anything about that particular study, but there's also a fairly low coverage study of major depression. And I think they decided on a threshold of 2. And the idea is that you have a whole bunch of cases and a whole bunch of controls. And you sequence random portions of both of these. Eventually you're going to get, across many, many people, you're going to get some information about every location in your cases in general, and some information about your controls in general. And so if an allele is much more common in cases than it is in controls, even though I've only done a couple of reads at every locus, or in some cases no reads at every locus, if there's an A allele and a B allele at a certain site, all I need to be able to say is that I'm confident that it's more prevalent in cases than in controls. And I think this was the central argument behind the design, at least of the major depression study that I'm thinking of. This was a study that came out of VCU. And the idea being that all we're interested in is frequency difference, and we don't need to confidently call the individuals in the data set.
I think in the case of the PSID, there were 2 arguments in favor of doing this over the SNP arrays. One was you get a lot more, you measure a lot more SNPs. It was like 50 million instead of 2 million for the SNP array. And even though it's different in different people, you can then use imputation to do a lot better in terms of getting similar information across different people. And then you avoid, the PSID has a lot of minority individuals, and so you avoid the problem with the Euro bias that Andrea was mentioning by taking essentially random draws of where in the genome you're measuring.
Cool. So that does raise the imputation question. The genotypes could be different for different people. But the SNPs that we actually have, I think Joel asked this when we were talking about SNP arrays, and I was saying that no, we have the same SNPs on everyone, but now that's not the case.
Yeah, so I think the company that did this for the PSID, GenCove, which is run by Joe Pickrell, they argue that you do this, that you get the data, but then they'll re-impute and re-impute over time as you get more and more people who are in the database, and so that those imputations can be more population specific, and so essentially you'll get more accurate data over time from that.
Oh, cool. I have not heard of that. That's pretty cool. Yeah.
So sort of that major depression study that you're mentioning, sort of low coverage study. I guess I'm just wondering, in terms of these kind of GWAS studies, there is a kind of bias associated with automatically pooling people and looking for collective. There's an underlying assumption that anything you get out of there that is collective is, so I'm just saying that there seems to be a bit of a circularity there, because first of all, like the allele frequencies themselves, there's a lot of noise in those cases, right? Because you have a lot of missing in the individual cause itself.
Yeah, and again, this is not something that I worked on closely, but I believe that there's also a paper walking through the statistical arguments. I think that paper came out of UNC for why this is okay, given the way that the technology works and given this study sample that they were proposing. I don't know all the details in that paper, but I do remember seeing it and being vaguely convinced. Okay. So you can also look at all your reads away and we can, yeah, I think I might have mixed these up. Yeah, so I want to talk about the exome now. So we talked about how the actual coding sequence of the genome is actually much smaller than the genomic sequence, and that we could refer to that coding sequence as the exome. So this is the protein coding business, and this encodes the majority of the functional part of the genome. And so if you were only interested in coding variants, you could sequence your exome and not your genome. And so this is a heck of a lot cheaper than doing the whole genome, but we require some additional technology. So how do we know which pieces are exons and which pieces are introns? And the answer is we use the existing annotation, and then we design these very specific probes called baits to bind to the sequence that we think is an exon and pull it out of solution so that we are only left with these coding bits of DNA. And that's done before you run any of your sequencing. And then the same technology applies. So once you've isolated the DNA that you're actually interested in, you run it through a flow cell, and you end up with an exome. So this is a picture that I borrowed from Elizabeth. I think it's a really nice idea that the whole genome is this whole apple and the exome is this very, very thin slice. So you can imagine your money would go further, but you're now not inquiring about regulatory variation. You're only inquiring about the coding sequence itself. You also run into problems, and there have been reported issues that these baits are sometimes not inclusive of every exon, that we maybe miss one, or that we are including something that's not actually an exon, but we think it is. The specificity of binding of these is also very important. So there's a lot of complicated chemistry that goes on there. So the idea here is that you have the best quality data for the exome. You have the lowest cost. And so lots of folks have been doing exomes. These are quite a bit cheaper than genomes. But again, this is a very small subset of the variation of human genetics. Okay, and Elizabeth has some really nice slides about how we can balance how deeply we want to sequence, whether we want to do genomes or exomes, or do we want to target a very limited set of genes. And this is going a little bit more towards the candidate gene approach, and all of these are considerations when you're designing a next generation sequencing study. So there are a couple of other applications to this. These kinds of things are starting to be used to query RNA, which we talked about. They're also starting to be used to query methylation and these kind of epigenetic marks. And this is an area of study that I am actively learning about, but I don't feel qualified to tell you about in any great detail. However, I can tell you about RNA-seq because that's the data form that I work on. And so this can absolutely be applied to RNA. So basically, it's the same procedure, but we create cDNA first, because remember we mentioned in the first lecture that RNA is not very stable in its single-stranded form. And RNA-seq provides a little bit different information about biology, right? If we're doing DNA sequencing, we are interested in where the variants are and what type of variation they are, how common they are, and those kinds of things. RNA-seq will query what's expressed, so the cell type matters, the time point matters, and the conditions matter. And then we can ask what genes are active, what genes are differentially expressed between conditions. This could be drug and control. This could be two different species, and then what are the splice junctions, and what isoforms might be present. And so all of these questions are questions that I focus on in my own research. And so just a cartoon, I can use this, of how this ends up looking. You might imagine that you have several genes on a transcript, and then there's some transcriptional landscape. There's some big glob of things being expressed, and I want to know how many transcripts of each thing there are. And so how this ends up happening is you have an RNA molecule, and we didn't talk about this at length on the first day, but when we transcribe things in general, there's a long series of repeated A's at the end of a gene. And so that's called a poly-A tail when you end up with RNA. And that poly-A tail, if you just stick a bead in your solution with a bunch of single-stranded T's on it, that will hybridize pretty nicely. This strand might break while you're pulling it down off the bead, but you have, at that point, captured a fragment of RNA. And so you can do this in a multiplexed way, and you end up with digested RNA fragments. So this is how RNA is captured in a cell. You end up with the digested RNA fragments, and you reverse transcribe them. There's several viruses that use reverse transcriptase, and that's sort of where we get the idea from, to make cDNA fragments. And then you replicate these up so that you have a whole bunch of them and run them through a flow cell just like it were DNA. And so you end up with paired end reads. These look sort of like Imperial fighters from Star Wars. But at the end of this whole process, you have your little paired end reads. Often the middle is much larger than the sequencing on the ends. Nope, wrong way. And then you have to map these. And this question of mapping in RNA is a little bit more complicated than the issue of mapping in DNA, because now I'm not just matching the sequence. I've already spliced out some introns. And so it's not just the human genome reference, but also the reference annotations of where splice junctions might lie. And there's quite a bit of probabilistic computing that goes on in inferring this. The best method, or the state-of-the-art methods right now, use 2 passes where they do an alignment assuming that we know the annotations of the splice junctions, and then they do it again, removing the splice junctions that we don't think are present. And all of that to end up with some aligned paired end reads. So once I know that these sequences have aligned to this gene, I can do that for all my other genes. And then we count. How many reads did I get from each gene? And then the standardization and normalization and interpretation of these counts is another whole active area of research and something that I work on. But in principle, this idea that I now have counts from different genes tells me which genes are active. And if I do this in 2 different conditions across several samples, I can ask which genes are differentially expressed between the conditions. And this is a big piece of how people assess expression.
So when someone talks about gene expression, like they're just referring to these numbers after this big process.
Yeah, if they're talking about RNA-seq data. So we talked about GWAS arrays. The array technology, this idea of having a microchip with single-stranded probes on it, actually was an expression technology first. And so you can imagine that's much, much easier in a way. Maybe I should have talked about this first. But if you have a microchip with a probe sticking off of it, and the probe is some unique sequence of the gene that has no variation in it or no known variation, then you get all of your transcripts sticking to this probe. It fluoresces, and now you have a measurement of how much of this transcript you have. And so that's what an expression array does. And the expression arrays, they were not as reliable as RNA-seq. In a lot of ways, this fluorescence technology, if you want to call a SNP, the only thing we're asking is it mostly A, mostly T, or some of each. When you want to call a level of expression, now you're continuous.
Because you look at like how brightly it fluoresces.
Yeah, it's just how bright is it. And across chips, you could see effects in time of day. You could see effects on the technician. And so that brightness required really a lot of normalization and standardization that sometimes squashed out signal. And so RNA-seq seems to be a little bit less technically buggy. And so even though the array technology was an RNA technology first, the SNP technology has been much more successful because the thing that we actually have to decide is easier to do given the raw data. Cool. So I asked about what genes are active and what genes might be differentially expressed. But we can also do things that we never could do with an array. We could ask about splice junctions. We could ask about isoforms. And so now we're going to reimagine the cartoon that I just gave you. And instead of 4 different genes, we're going to have 4 different exons. You can imagine that in this, we're still in biology land now. I haven't done anything technical yet. And that these are the possible mature transcripts floating around in my cell, and these are the transcripts that I want to know about. We still have to do RNA shearing. We still have to do cDNA and make fragments for sequencing, but now some of these junctions that are not present in the original sequence, so they're not present in the reference genome, are present in our exons. So only a couple of fragments have exon-exon junctions, and that's okay. So we take those, we sequence them, and some of the junctions are going to end up in this middle place where we don't actually know where that is, but we will actually go over the junction in a number of reads, especially if our sequencing depth is high enough. And then we have to align these back, right? We don't actually know which exons those go to, so we have to align them using what we know from the annotation, and if we have enough sequence context. And one of the problems I spent a while working on was how much sequence do you need on either side to be sure that you're calling the right exon and not just making a guess. So this question of isoforms, sort of a last question, is actually quite difficult with short reads because we can tell you how many junctions of what are there, but we don't necessarily know which version of exon 2 connects to exon 1 and exon 4, and which version of exon 2 connects to exon 1 and exon 3. So that is still a very active problem that lots of folks are working on. Cool. So we talked about RNA-seq. Just a couple of words on protein, and again, this is really starting to get outside of what I think about and what I do. But my understanding of protein is that it is much harder to measure, and that simply sequencing the protein is not enough. That capturing specific proteins usually requires an antibody, and that these antibodies have to be specific, and you have to know what you're looking for. And so the level at which we can multiplex our protein capture is far, far behind the other omics technologies. And very recently, these were done one at a time. There are lots of measurements that you want to look at. You want to look at total amount, you want to look at weight, 3D structure, and the sequence of amino acids. And there are starting to be technologies that multiplex proteins, but these are on the order of thousands, whereas with SNPs, we are on the order of millions. And so these technologies are starting to become available, but they're still very early days in comparison to the other omics that we've talked about. And I think that is all I have for you. Are there questions about this stuff or about anything else?
Yeah, so for people who talk about metabolomics, where does that sort of fit into this?
Okay. So metabolomics, I wish Claire was here. We have some members of our group that work on metabolomics specifically. My understanding is that metabolomics are anything that can be metabolized. So that's forms of cholesterol, and these are other biological molecules.
Of proteins?
Some of them are, but not all of them. So these are specific biological molecules with specific functions, and the metabolite profiles are basically a kit of chemistry that will attack each one of those things and attempt to quantify each one of those things individually. But my understanding is that the chemistries involved for each one of the metabolites can be very different depending on what type of molecule it is. And again, that's my understanding. If somebody knows better, please chime in. Yeah.
So you mentioned this earlier, but that a lot of these technologies that go beyond measuring genetic variation are likely to be of less interest to social scientists because we're mostly interested in what's going on in the brain, and you can't get large samples for that. Are there, so I guess the exception is immune function. To the extent we're interested in health and immune responses, that's probably something you can get from non-brain tissue. Are there potential ways that you might be able to imagine measuring like like taking a draw of brain tissue from a live person and potentially getting some of this information?
Yeah, and I guess I don't know enough about the medicine behind brain biopsy to answer that specific question. I will say that there are several biobanks of post-mortem brain tissue that are starting to become available. The GTEx consortium, being the most notable, has quite a collection of brain tissue that is all post-mortem, but in many of the samples, the post-mortem interval is quite small, and so these are pretty fresh brains. They do 13 different brain regions. A lot of folks are doing single cell work on neurons, and so if you had a process that you were interested in, some kind of stress process, some kind of process that we understand produces some kind of physiological effects in humans and has a biological pathway that you can go after, and you know the cell type of interest, then there's a lot of experiments in a dish that could be potentially interesting. And then also, if there's a physiological response as medicine and our understanding of biology progresses, there may be some cases where we can say, well, there's a change in the brain, and after that change in the brain, we observe some change in the blood. Then you could start looking at blood or some sort of proxy tissue instead of directly looking at neurons. Yo.
Actually, I read that there is a single cell experiment which did in fact, it was essentially trying to test for patients who had epilepsy, trying to do temporal cortex kinds of surgeries where they would essentially scrape out certain things by sort of removing the top layers and going deeper. And the idea is they use that for subsequent sort of single cell analysis. So that it is from live patients under certain types of obviously clearly very invasive interventions. Those cases, but obviously the cases are probably small. There are, I think, brain tumor samples also in a similar fashion, but those are extreme cases under which people
Yeah, and I think it's still an open question in terms of the types of in-the-dish neurons, these induced omnipotent stem cell or pluripotent stem cell models, that how close those are to actual in vivo human neurons. And I think only time will tell whether those could be used for the kind of study that you're imagining. If there are no more questions, then I think it's lunchtime.